Hello students, today we can see another pathway of photosynthesis, that is the C4 pathway. What are C4 plants? The plants in which the first carbon dioxide fixation product is a C4 acid, that is oxaloacetic acid, are called the C4 plants. Normally, the plants that are adapted to dry tropical regions follow C4 pathway. Plants like sugarcane, maize, sorghum, euphorbia are C4 plants. Even though they follow C4 pathway, the main biosynthetic pathway by which they prepare their food is C3 pathway or the Calvin cycle. Okay. Now, what is special about the C4 plants? Why the C4 plants are special? They are having a special type of leaf anatomy, that is Kranz anatomy. They can tolerate high temperatures, that is they can withstand high temperature. They show response to high light intensities. Even C3 plants also show response to light intensities, but when high light intensities are there, up to a certain extent they can withstand that light intensity and they can perform photosynthesis. Beyond that, when the C3 plants are subjected to high light intensities, what happens? Photooxidation of chlorophyll A occurs. Okay, but here the C4 plants can show response to high light intensities. They lack a process called photorespiration. Photorespiration means what? Respiration in the presence of light, isn't it? They take oxygen and release carbon dioxide in the presence of light. So that photorespiration will not be seen in C4 plants. Then they have greater productivity of biomass. That means a more amount of organic matter can be produced by the C4 plants. So these are some of the specialities of C4 plants. One special feature of the C4 plants is their leaves show Kranz anatomy. What is Kranz? Kranz means a wreath. That is, their bundle sheath cells are arranged in a wreath-like manner. Wreath-like means you can see here, that is somewhat in a circular manner. The bundle sheath cells will be arranged. So these are the bundle sheath cells. So they are arranged in a circular manner around the vascular bundle. So here you can see the vascular bundles. What is vascular bundle? Vascular bundle is formed of xylem and phloem. Okay. So around the vascular bundle, the bundle sheath cells are present. And these are large cells and they form several layers around the vascular bundle, which gives a characteristic wreath-like structure, and that's why this anatomy is said to be as Kranz anatomy. Okay. And these bundle sheath cells are having chloroplast in them. You can see here it is green in color. So it is due to the presence of chloroplast. The bundle sheath cells are having chloroplast in them, and the cells are surrounded by thick cell wall, and it is impervious to gaseous exchange. That is, it will not allow the gas to pass through. Okay. And the cells are arranged very close to each other without any intercellular space. So you can see there is no intercellular space between the cells. The cells are closely arranged. Okay, so this is the characteristic features which is seen in the leaf of C4 plants. But if you take the C3 plants, they do not show such a Kranz anatomy. Their bundle sheath cells are not this much bigger. They are little smaller, and normally they do not have chloroplast in them. Okay. Now we can see how the C4 pathway of photosynthesis takes place. It is also said to be a Hatch and Slack pathway because it was given by M.D. Hatch and Roger Slack. It occurs in the C4 plants. And in C4 plants, the C4 pathway occurs in mesophyll cell as well as the bundle sheath cells. In C3 pathway, it occurs only in the mesophyll cell. But here in C4 plants, the C4 pathway occurs both in the mesophyll cell as well as the bundle sheath cell. The primary carbon dioxide acceptor in C4 pathway is a 3-carbon molecule called phosphoenol pyruvate. Okay, so you can see here it is the phosphoenol pyruvate which is the primary acceptor of carbon dioxide. Here you can see the carbon dioxide is present in the form of bicarbonate, isn't it? Now, how that bicarbonate is formed, you might have studied in respiration that carbon dioxide combines with water to form carbonic acid. And which is the enzyme involved? It is carbonic anhydrase, and this will be again splitting into bicarbonate and H plus by the same enzyme, that is carbonic anhydrase. Okay, so when carbon dioxide is present inside the cell, when it enters inside the cell, immediately it will be hydrated and it forms the bicarbonate. Okay, so this bicarbonate will be accepted by this phosphoenol pyruvate in the presence of the enzyme PEP carboxylase. Okay, phosphoenol pyruvate carboxylase helps in the fixing of carbon dioxide, that is in the form of bicarbonate with the phosphoenol pyruvate. As a result, what is formed? A C4 acid is formed. What is that C4 acid? The C4 acid is nothing but oxaloacetic acid, OAA, that is oxaloacetic acid. Okay. Now, this C4 acid or this oxaloacetic acid can be then converted into other 4 carbon acids like malic acid or aspartic acid. How malic acid is formed? OAA will be reacting with NADPH. As a result, what will be formed? Malic acid will be formed. Okay, malic acid plus NADP will be formed. It is catalyzed by the enzyme dehydrogenase. Okay, so OAA or oxaloacetic acid, which is formed inside the mesophyll cell, can be converted into malic acid, or even it can be converted into aspartic acid also. How it will be converted into aspartic acid? Here OAA will be reacting with an amino group, okay, and NADPH. As a result, again the same enzyme, okay, what is that? Dehydrogenase enzyme. As a result, what will be formed? Aspartic acid will be formed, okay, aspartic acid plus NADP, okay, and water will be formed. Okay, so this is how aspartic acid is formed from oxaloacetic acid. So we can say aspartic acid is formed by amination. Amination means one amino group is added to it. So it will be forming the aspartic acid. Is it clear? As a result, water and NADP also will be released and it is catalyzed by the enzyme dehydrogenase. So this oxaloacetic acid is converted into malic acid or aspartic acid in the mesophyll cell itself. Okay. Now these acids are transported to the bundle sheath cells. Okay, then it is transported to the bundle sheath cell. Okay, so here that C4 acid is transported, that is the malic acid or aspartic acid is reaching now the bundle sheath cell. Okay, now what happens in the bundle sheath cell? Now the malic acid is decarboxylated into pyruvate and carbon dioxide in the bundle sheath cell. Okay, so what happens in the bundle sheath cell? The malic acid will be decarboxylated to form pyruvate and carbon dioxide. Okay, so malic acid, we can take a malic acid plus NADP, okay, gives malic enzyme will be malic enzyme. It gives a pyruvate plus CO2 plus NADPH. Okay, so now what is formed? Again carbon dioxide is formed inside the bundle sheath cell. Okay, so malic acid will combine with the NADP to form pyruvate carbon dioxide and NADPH, and here again what is formed? Carbon dioxide is formed. Okay. Now what happens to this carbon dioxide which is released as a result of decarboxylation of malic acid? It will be involved in the Calvin cycle. It takes part in the Calvin cycle. Why Calvin cycle occurs in the bundle sheath cell? Because RUBP is present in bundle sheath cell and Rubisco enzyme is present only in the bundle sheath cell. Rubisco enzyme is absent in mesophyll cell. In mesophyll cell, PEP carboxylase, that is phosphoenol pyruvate carboxylase enzyme is present in mesophyll cell, and that is absent in the bundle sheath cell. Okay, so now the carbon dioxide which is released will be accepted by the RUBP. So the secondary acceptor of carbon dioxide in C4 pathway is RUBP. Okay, which is the primary acceptor of carbon dioxide? It is phosphoenol pyruvate, which is present in the mesophyll cell. Okay, so here the carbon dioxide will be accepted by RUBP in the bundle sheath cell, and it will be converted into 3-phosphoglyceric acid, which is catalyzed by the enzyme Rubisco. Okay, because Rubisco enzyme is present only in the bundle sheath cell. Now this 3-phosphoglyceric acid will undergo a series of steps in the reduction process, and at last a glucose molecule or carbohydrate will be synthesized. Okay, so due to the decarboxylation of malic acid, carbon dioxide and a C3 acid is formed, isn't it? What is that C3 acid? It is the pyruvic acid or pyruvate. What happens to this pyruvic acid? It will be transported back to the mesophyll cell, where it will be converted into phosphoenol pyruvate by utilizing ATP molecule. Okay, so this C3 acid, that is pyruvic acid, will be converted into phosphoenol pyruvate in the mesophyll cell, and in the mesophyll cell again this PEP or phosphoenol pyruvate will be ready to receive another carbon dioxide in the form of bicarbonate. Okay, and again the cycle can take place. So from this you can find out that even though we say it as C4 pathway, the main biosynthetic pathway by which the food is synthesized is the C3 pathway, and that occurs in the bundle sheath cell. Okay, so this is how C4 pathway takes place in C4 plants. Okay. Now we can see the process that creates an important difference between C3 and C4 plants, that is photorespiration. What is photorespiration? It is the uptake of oxygen and release of carbon dioxide in the presence of light. To understand this process in a better manner, we can see the first step that occurs in Calvin cycle or the C3 pathway. So which is the first step in C3 pathway? It is the fixation of carbon dioxide by RUBP, isn't it? So here what happens? RUBP combines with carbon dioxide to form two molecules of 3-phosphoglyceric acid, which is catalyzed by the enzyme Rubisco, isn't it? What is Rubisco? Rubisco is the most abundant protein which is present in the biosphere. Rubisco is an enzyme. The full form is ribulose bisphosphate carboxylase oxygenase. And you know that all enzymes have active site in which the substrate will be binding, isn't it? Rubisco is also having its active site which can bind to both oxygen and carbon dioxide. So it is named as ribulose bisphosphate carboxylase oxygenase enzyme. Okay. This Rubisco is having a greater affinity towards carbon dioxide than oxygen when the concentration of oxygen and carbon dioxide is more or less equal. Okay, you think of what will happen if suppose if it is having more affinity towards oxygen, then oxygen will be combining, and as a result combining with the Rubisco, and as a result what happens, carbon dioxide fixation can be affected, isn't it? It will be decreased, and the food synthesis will also be affected. Okay, so normally or generally Rubisco acts as a carboxylase enzyme because it is having greater affinity towards carbon dioxide than oxygen. Okay, now in C3 plants some oxygen binds with Rubisco. That is, when the concentration of oxygen is more, then some oxygen can bind with the Rubisco, because Rubisco now will act as a oxygenase enzyme than carboxylase enzyme. Okay, and because of that the carbon dioxide will be decreased. Okay, generally RUBP combines with the carbon dioxide to form water. 2 molecules of 3-phosphoglyceric acid, isn't it? But when the concentration of oxygen is more, when Rubisco acts as an oxygenase enzyme, then RUBP will combine with oxygen. Instead of combine with carbon dioxide, it now combines with oxygen. As a result, it forms 1 molecule of phosphoglycerate. Phosphoglycerate, we can also say it as the 3-phosphoglyceric acid. Okay, so 1 molecule of 3-phosphoglyceric acid and 1 molecule of phosphoglycolate will be formed instead of 2 molecules of 3-phosphoglyceric acid. Okay. So if Rubisco is acting as a carboxylase enzyme, then RUBP will combine with carbon dioxide to form 2 molecules of 3-phosphoglyceric acid. But when Rubisco acts as an oxygenase enzyme, then RUBP combines with oxygen to form 1 molecule of phosphoglycerate or 3-phosphoglyceric acid and another molecule of phosphoglycolate. Okay, so here no sugar will be synthesized, no ATP will be synthesized, or no NADPH will be synthesized. Okay, so during photorespiration, sugar, ATP and NADPH will not be synthesized. Okay, instead of that carbon dioxide will be released with the utilization of ATP. How carbon dioxide is released? This phosphoglycolate will be losing its phosphate group and it becomes glycolate. Okay, this glycolate will now oxidized so that glyoxylate will be formed. Okay, and this glyoxylate will be now converted into the amino acid glycine. Okay, and when two glycine molecules interact with each other, it form another amino acid called serine, and carbon dioxide will also be released. Thus the carbon dioxide will be released, and during this process it will be utilizing ATP. Okay, so in C3 plants, photorespiration occurs. As a result, carbon dioxide fixation will be affected because carbon dioxide will be released into the atmosphere as a product of photorespiration. Okay, now on the other hand, you take the C4 plants where this C4 pathway occurs. It occurs in the mesophyll cell and in the bundle sheath cell. Okay, so here no photorespiration occur. Okay, now why photorespiration will not occur here? Because in the mesophyll cell, Rubisco enzyme is absent and only PEP carboxylase enzyme, that is phosphoenol pyruvate carboxylase enzyme is present. So the carbon dioxide which was entering there will be utilized, or it will be fixed by the PEP or phosphoenol pyruvate to form a 4-carbon acid, that is OAA. What is OAA? It is oxaloacetic acid. It is a C4 acid, isn't it? Now this C4 acid will be converted into an other C4 acid like malic acid, aspartic acid, which will be transported into the bundle sheath cell. Isn't it? In the bundle sheath cell, what happens to this malic acid? It will be decarboxylated to form carbon dioxide and the C3 acid. So this carbon dioxide can be fixed, or it can be used for the formation of glucose in the pathway called C3 pathway, because in this bundle sheath cell, what is present? Rubisco enzyme is present. So this Rubisco enzyme is absent in mesophyll cell. In C3 plants, it is present in mesophyll cell, and the mesophyll cell can be easily subjected to the oxygen, isn't it? But here, the mesophyll cell, which is present towards the outer surface next to the epidermis, is not having the Rubisco enzyme. So this Rubisco enzyme cannot act as oxygenase when the oxygen concentration increases, because it is present in a protected region, that is in the bundle sheath cell. Isn't it? So in the bundle sheath cell, only which concentration will be increasing? The concentration of carbon dioxide will be increasing. How it increases? Whenever malic acid or aspartic acid is decarboxylated in the bundle sheath cell, it will release carbon dioxide. So the carbon dioxide concentration increases in the bundle sheath cell. So when carbon dioxide concentration increases, automatically Rubisco enzyme will be acting as what? Carboxylase enzyme, and it can help in the fixation of carbon dioxide. Okay, so thus we can say photorespiration does not occur in C4 plants. Okay, and in C3 plants, this photorespiration occurs because Rubisco enzyme is present in the mesophyll cell. Okay, so in C4 plants, Rubisco will be functioning as a carboxylase enzyme, and its minimum activity will be seen as an oxygenase enzyme. Okay, so now from this we can say that C4 plants lack photorespiration. Because of that only, the productivity and yield is better in C4 plants when compared to the C3 plants. Okay, in C3 plants, photorespiration also occurs. So the carbon dioxide fixation will be less. So the amount of organic matter or the food which is synthesized will be less. So the productivity and yield will be less in C3 plants when compared to the C4 plants, because in C4 plants, which process will not be taking place? Photorespiration will not occur. So the productivity and yield is better in C4 plants than the C3 plants. Okay.